Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. I'm very excited to be here today with Justin Porcano, who's the co-founder and executive director of Save Sight Now. They're a nonprofit working to find treatments for Usher syndrome type 1B, which is a rare genetic condition that causes deafness at birth and progressive vision loss. Justin got involved in this after his daughter Leah was diagnosed with USH1B, and Justin and his wife Rosalind started the nonprofit to fund and accelerate research into therapies into the condition. They host an annual fundraiser, which is coming up, and they've done a lot of incredible work, both on their own and also partnering with Foundation for Fighting Blindness and other partners. So Justin, thank you so much for joining us and looking forward to the discussion today.
Yeah, absolutely. Thanks for having me, Patrick.
I'd love to just start with your story. If you could take us back to when you decided that you were going to found Save Sight Now and transition from being a father of a child that's affected by this condition to someone who's really taking a leadership role in driving research forward.
Sure. 2018, we had our daughter Leah. She failed her genetic hearings or her hearing screening in the hospital. So thank God for those screenings, by the way. They're super important. That raised flags immediately, which set a bunch of things in motion. Leah would see an audiologist 2 weeks, at just 2 weeks old, confirm that she was profoundly deaf. After that, we submitted for genetic testing. We got the results back 5 months later, which were pretty devastating for folks who don't know. Usher syndrome type 1B Usher syndrome in general is the leading cause of inherited combined blindness and deafness. USH1 subtype is the more aggressive form. Leah has Usher syndrome type 1B, which basically means you're completely deaf from birth. You have vestibular issues as well. So you have very poor balance and you rapidly lose your vision. Typically, the path is legal blindness by high school. And when we got that diagnosis, I think it took a couple of weeks for us to recover from that. And then I immediately started asking around, well, what's going on? Is there anything happening in this space? And Rosalind, my wife, and myself are realists, right? We weren't going to basically make a major life pivot unless we found that there was something, a little glimmer of hope. So we did actually found out there was a clinical trial that was in limbo called USHStat, I believe. That turned out to be a failed trial using lentivirus, but what it did was that it opened our eyes. It said, not only are people interested in this, but people are actively working on it, trying to find a treatment. And that was all I needed to hear to say, okay, it's time we get involved and see how we can help accelerate this. And that was kind of the moment where we decided to start doing something. First thing I wanted to understand was, well, what are the biggest hurdles or roadblocks to getting into clinical trials? What do researchers want? What's gating them? And at that point, I'm an industrial designer. I have no background in this space. I was working full time, as was my wife. We didn't know how to start a company or didn't even know what to point it at. We reached out to the Foundation Fighting Blindness. They're the largest funding organization for inherited retinal diseases when it comes to funding research. We wrote up a little agreement that said, hey, listen, we don't know what we're doing, but we think we can raise money. If we raise money, can we partner with you to make sure that it's restricted for USH1B research? They agreed to that. We raised a ton of money. We learned how to raise we learned how to do everything here.
How much did you raise?
Our first year, we raised about $250,000.
Wow.
Mostly off like a GoFundMe video. We have a very talented friend, Jay Redman, who's a director who does a lot of documentaries, which was perfect. So he put together a really nice video for us. And that's kind of how we didn't just launch Save Sight Now, but it was kind of like us coming out to our friends and family what was
Right.
going on with us, because that's a big deal too. There's always this balance of like how much of yourself do you put out there and expose, especially with a young child. And we figured if we're gonna have an impact, we have to put ourselves out there. And we've always been very private people. So that was challenging for us. But, we decided not to let that get in the way of progress.
That is amazing. And at the time, you mentioned wanting to get to the bottom of what are the big bottlenecks into research. And just to give people background here, and please correct me if I'm wrong, the genetic cause is MYO7A gene. It's autosomal recessive, and it's basically a stereocilia, small hair structures in the ears, in the inner ear, which affects both the hearing and the vestibular function. And then I think the protein also has a function in the eye, which contributes to the progressive blindness. Obviously, the genetic cause was well understood at the time. What were the challenges? Were they around understanding the mechanisms? Was it therapeutics? Was it a combination of those? Maybe you can take us back to where it was then, and then obviously we'll talk a little bit about where we are today.
Yeah, I think basic biology in the rare disease space is always a challenge. I don't think most diseases in this space have a complete understanding of what the cells are doing or how they're interacting within their own environment. That wasn't the biggest hurdle though. Like if you talk to researchers, if you're doing a gene replacement, you don't always need to know all. You just need to get a healthy copy of a gene in the right
Yeah.
location, right? And like you can theoretically skip understanding the nuances of all the biology. So the biggest challenges to a therapy were myosin 7A is a large gene. You can't fit it in a single AAV, being one of the biggest ones. Now you got to figure out, okay, large gene delivery systems, right? I think a lot of people have this issue. The other one, we were lacking a good animal model. While there is a USH1B myosin 7A mouse called the Shaker 1 mouse, mice in generally don't structurally, myosin 7A doesn't structurally live in the same location within photoreceptors, I believe, within mice. There is a structural incompatibility with the human eye where
Huh.
it's not a great model, which sucks because mice are cheap and they're easy to reproduce. So you always want a good mouse model. So we didn't have I mean, you can do some early proof of concept stuff in this mouse model, but I think when you start climbing the kind of the therapeutic ladder as you get closer towards a treatment, you want something with a little bit more fidelity. So we had an animal model issue, and then from kind of a regulatory point of view, I don't think we ever had any good clear endpoints defined. And that's what as I went to all these meetings and conventions, you talk to biotech and pharma, they're like, we wouldn't touch this unless you can tell us what endpoints are. We're not even think about investing in a treatment without endpoints. So those became our kind of like three targets was large animal models or a legitimate animal model. It doesn't even have to be a large gene delivery, or if we're not doing a gene therapy, something else that avoids that. And how do we get to clinical endpoints that the FDA will recognize? Those became our really three focal points. And our logic, and this comes from David Gam, kind of a leader in the cell space was, just build the tools. Like you don't, as a small organization, it's not like you can raise 20, $30 million. But if you build the tools that attract the researchers and you help them solve their hurdles, you'll just get a lot of attention and you'll get more interest around them wanting to use their strategies on your gene.
Yes, that's right. And so for you, fast forward to today, which of those have progressed the most, and where's the biggest bottleneck still?
I'd say we've invested a lot in animal models. There is a H1B pig cohort in Germany right now where they were found a naturally occurring, which is wild, H1B porcine model. It has the vestibular issues because they just run in circles. So you can tell really quick that there's something going on. Then they did the auditory phenotyping that has the same auditory. And then now they're in the process of doing the retinal phenotyping. And they believe that it's going to prove to be a really valuable model. We already have researchers who are like, how quick can I get my hands on that model? We've also invested a lot in the cellular space doing iPSC lines because I think you want that broad spectrum of testing from less expensive and quick to a little bit higher level fidelity and, to give you a little bit more confidence within an animal model. Like it's one thing to test cells siloed in a culture. It's another thing to test cells next to all the other cells they interact with in their real environment. And we've also invested at OHSU to create a non-human primate where we basically you edit myosin 7A in the kind of the embryonic stage so that you give birth to a non-human primate that has USH1B. And the amazing talented researchers at OHSU were able to create one non-human primate currently, and they're trying to kind of replicate those tools and protocols so that that can become a replicable process for them.
Oh.
And
And yeah,
sorry, one last thing, Patrick. Yeah, on the clinical endpoints, we're currently funding the Unirare project, which is we've got 40 cohorts in a USH1B natural history study, and we've also funded work at the Institut de la Vision, where they also have a natural history study going on.
wow. Tell me about how you get up to speed on all these. You mentioned at the top that you have a background in industrial design. You know, you're clearly smart and a fast learner. How like what do you do literally? I'm thinking about the families in the audience that are thinking about this or anyone who wants to learn about something you know, you have the motivation, but like do you these days do you use AI? Do you read papers? Do you talk to experts? What do you do to jump up to speed so quickly?
Yeah, I mean, I'd say funny enough, industrial design has set up a framework or a methodology for me to approach a problem.
Hmm.
And regardless of what I'm designing, whether I'm designing a cell phone or a piece of furniture, there is a thought process that goes into understanding what the current challenges or problems are, identifying those, and then figuring out how to address or overcome those. And you just apply that same methodology to anything basically. And that's what I did here. The first step being like, let me talk to researchers and I want to hear from them what's gating them, what would kind of like open up doors and speed up a path to a clinical trial. And that's when we identified delivery systems for gene therapy and lack of animal models and endpoints. So then I said, then the process is like, okay, well, let me go find who I want to be my sounding board. Who is my scientific advisory board here that are specialists in each one of these spaces? And I just dug. The funny, like, my education has happened at the transition from no AI to AI. Like, I would read through papers with like a dictionary and a thesaurus, and
Yeah.
I would have to figure out what every other word meant. And now I literally just copy and paste a whole publication into like chat, and I say, break this down for me in layman terms so I can understand it. And it's wild how quick you can like upskill or educate yourself, at least from a basic point of view. So really quickly, between speaking to some of the researchers are so open and welcoming, like especially the researchers who are also clinicians, really are open and give you their time. And that's just how I learned, talking to researchers and reading papers.
Yeah, that's amazing. And so then coming back, because the reason I wanted to ask, because your framework is yeah, it's very organized, and I love the way you break down the problem, look at the bottlenecks, and you just start to work backwards from there, right? If we think about therapeutics, where there's a very different picture today than there was in 2018, right? You have a number of gene therapy companies. There's a lot going on. I'd love to get your perspective on that. What are the most promising things that are going on, and what are the big things that have changed in the last couple of years from that perspective?
Yeah, I'd say so because we are in the too large of a gene to deliver in a single AAV space. We're mostly interested in anything that can kind of overcome that challenge, whether that is like a lipid nanoparticle or a synthetic nanoparticle. I think there's promise in that space. For USH1B, there's a clinical trial happening in Italy right now for a dual AV approach. There are other companies working on dual AV approaches. Those excite us. I mean, dual AV is,
Can you
biologically,
explain dual AAV for people who aren't
yeah.
familiar?
Sure. So you are, because a gene is too big, you're basically cutting it in half. You're putting it in each part in its own delivery package, same package that you deliver all of them in, and you inject that into the back of the eye, and that those ends or parts find each other, recapitulate, form a gene that then produces a functional protein. So you're basically just cutting it in half to reduce the size. That's a dual AV approach. There are a couple companies there, so we're excited about that. There's what's called REJ, RNA end joining, which is kind of similar to where you're on an RNA level, which I don't know is more attractive, I think, to a lot of people because you're just rewriting the instructions, and I honestly, I don't know too much about that technology. That I think that's really one of the newer technologies. I think even newer than dual AV. And I guess you're splitting the gene up into several pieces and then really delivering them again because in those pieces because you have they're too big to deliver as a whole. And then you're joining them at the RNA level, like at the ETN level as well, or NTN level. That holds a lot of promise too. So I would say nanoparticles, new things like REJ, dual AV in the gene delivery space. And I think there's also a lot going on in retinitis pigmentosa that is the kind of the phenotype or the rationale for the loss of vision with USH1B. But but it's also an oxidative stress problem, and there's a lot going on with antioxidants that I think are showing some progress where you could at least give a patient antioxidants, providing you get enough cellular uptake and slow down progress retinal degeneration and open up your therapeutic window. The problem, Russ, this is a race against time. So
Yeah.
if we can do that now and open our therapeutic window instead of to 5 years to 20 years, wow, think of the time you can buy by doing so. I'm excited about what's happening, I think, in the drug repurposing, small molecule kind of space as well, too.
Yeah, fascinating. You maybe you can talk a little bit about this timing point because we've had Lawrence Reed from Decibel Therapeutics on long time ago. They were eventually acquired by Regeneron, and they've had a very successful gene therapy for otoferlin related genetic hearing loss. And that is this has me thinking about we have the multi organ system issue here, and I suspect you're going to have to go really early to intervene on the hearing because you mentioned it's effectively from birth, right? So I'm this is going to be a broad question, but how do you think about newborn screening programs and also relating the therapeutics to the intervention window? Because naively, the earlier and perhaps obviously the early we go, the better, but that's a central challenge when we don't have widespread newborn screening.
Totally. I mean, even doing genetic testing in pediatrics is just a major hurdle in general. I think for USH1, so if you're USH2 or USH3, you're born with hearing. You just have hearing loss, and you can rescue those hair cells, the hearing cells in the cochlea. For USH1B, you have to be kind of embryonic doing that. You have to, because by the time our daughter was born, those hair cells are they're completely destroyed. Typically, hair cells stand up and they kind of like
Right.
form like a wave right in the ear, and that's what's sensing the sound. If you look at images of hair cells from USH1B, they are laying flat, crossed over. They're kind of unrescuable at that point. You can rescue vestibular though, which is something we learned later in life. I don't think we have a final number, but there's been a couple great Heinrich out of Kansas City. He rescued not only hearing in mice, but he also rescued balance, which I
Wow.
think isn't it's an overlooked sensory for when we think hearing and vision, right? But when you pile on balance on top of low vision, quality of life and mobility becomes significantly impacted. So the fact that someone actually, I think they partner with Medell, who's a cochlear implant company, who would like to focus on restoring vestibular function. I think when it comes to hearing at this point, it's a time of intervention. I don't think hearing cochlear implants,
Yeah.
that's the way to go for kids with USH1B. For vision, yeah, as soon as possible. Because so for USH1B, like my daughter's not completely night blind and she's 7. So, sun goes down, she can't see anything like in kind of dim lit rooms.
Yeah, that makes sense. The underlying biology is really important here, right? In terms of how you intervene and where and what you can do. So what does next 5 years or so look like? We talked about the bottlenecks before that we're tackling. If you break it down today and say we're going to focus our effort in these couple of places, it sounds like you made some really great progress on the model systems. Where do we go from here?
We're, so we like I mentioned previously, we were partners with the Foundation Fighting Blindness for 6 years, and that was an amazing partnership. I can't emphasize that enough and how much we've learned from them. We've recently just become our own independent nonprofit and are in the process of building that 3 to 5 year roadmap. We're prioritizing speed. What can we do fast? What can we do we can afford? And what can we do to extend our therapeutic window? Those are our primary metrics and goals. And we have a couple of new families on board now that we've gone independent. We have the potential right now to annually raise $1 million and sustain that over the next 10 years. So $1 million a year can get you somewhere fast. We also have potential investors on board just through friends who said, listen, if you find a path you want to go that makes sense to start your own company, if you want to license something that another company is going to shelve, we're here to back that. So we are exploring everything from either supporting biotech companies who want help de-risking an investment in order to bring it into clinical trial. We're here to fund academia and researchers who have a hint or a path or relationship to pharma or biotech towards a clinical trial. I think our thing right now is that if we're prioritizing one thing, it's that we're not funding basic biology for the sake of funding basic biology right now. We need to see a clear path to clinic and understand how all the dots connect because of the time issue. So in the next 5 years, I hope to be in clinical trials with something, whether that is a small molecule, an ASO, or a drug that's been repurposed and it's maybe tweaked or slightly engineered. I think that's the quicker development path than maybe a gene replacement. So I think we're looking at it in parallel efforts. What can we do in the immediate that can slow degradation down? And then what can we be thinking about that has maybe a little bit of a longer runway that can permanently resolve retinal function loss?
How did you go about making the decision to go independent? Because you have this roadmap that you can take and build independently, but also, like you mentioned before, Foundation for Fighting Blindness is incredible organization, and they're doing amazing work on genetic testing access and all kinds of things. And obviously you keep that close link. But I'm just curious about how you made that decision, because probably a lot of parents who might be in this situation are thinking about, it is really helpful to get off the ground to partner with somebody who's at scale, but then at some point you maybe make that decision where it makes sense to become a little bit more independent.
Yeah, and that's exactly right. I think getting off the ground, it made a ton of sense working with the FFA. Also, the education and the exposure to their scientific advisory board was, just invaluable. We came to this decision when I think a couple things that we wanted a little more autonomy around, and that was the ability to approach pharma and biotech and support them directly. When the ecosystem we were working in, the funding ecosystem with the FFB meant we could only fund academic institutions. We couldn't actually work with some of these companies. The other thing is that they are on a annual cyclical grant cycle. And we wanted to be more, we like if something came up, I didn't want to wait a year to fund it. I want to fund it immediately. So I think having that flexibility to fund work as it comes up on a rolling cycle rather than an annual static one, those are the two biggest things. We wanted to work directly with companies and biotech, and we wanted to be able to fund work immediately.
Yeah, that makes complete sense. What is missing that you're looking for help from others on? Is it, funding? It sounds like you've got an amazing fundraising apparatus, and there can always be more of that. But from a infrastructure perspective, what's hard? Like is it hard to run natural history studies still and expensive? Is it hard to build these connections with biotechs? If you thought about one thing that could help, my thesis is that the more we can help groups like yours to build that foundation, like you mentioned before, and help it raises the waterline for biotech and pharma to pick it up. What is missing there to enable this to happen across every rare and ultra rare disease where there's a parent who has a vision like you have?
I think I think patient organizations are undervalued in a lot of organizational eyes, where I think if there was a closer partnership or collaboration between patient orgs and biotech, I think biotech keeps like they have hands like they keep you at a distance, which is odd because the access that we have to patients for them for clinical trials, the input that we could have around clinical endpoints, which is why we do like a PFDD with the FDA. But like, if they were to orchestrate, I think that a little bit more, like we're working with the FFB to do this talking session with the FDA. But like biotech, if they really wanted to de-risk some of these things, they'd go and fund some of their own clinical endpoint work. Again, we're in the rare disease space, right? So it's like I can imagine that there's this constant, is this worth are we going to see a return on our investment? How big is the population, right? I think once you get over that hurdle and you decide it's a path you want to go down, I think where a strong need is stronger partnerships and relationships between patient organizations and biotech companies would help both parties. I think you'd see research accelerate because I'm constantly going to biotech companies who are slightly interested in H1B or had a large gene delivery system I think is applicable. And I'm saying, listen, would $1 million a year help you in access to patients? I have an animal model I created. Would you like to use that? Like, and I don't think they often take us for real, like them seriously. And I'm like, No, we're serious.
Right.
Here's a pig model. Here's 50 patients who were waiting for a clinical trial and all their contact information. And here's $1 million non-diluted. Will that help de-risk this program? And I think biotech needs to start taking patient orgs like that more seriously.
Yeah, I think you're absolutely right on that. Something that I hear sometimes is also that biotechs don't want to let patient organizations down if they, if they lean in and spend some time and then they exit, it leaves it leaves a bad taste. And obviously if you do it incorrectly, it can, but it seems like that's worse than not engaging to begin with, right? And chasing it down.
Exactly. I mean, I think if you've been in this long enough as a patient and parent, you're going to get used to the letdowns and the
Right.
failed trials and the researchers picking another gene over yours. So,
Yeah. So the
yeah, it,
answer to
it,
the biotechs out there is we've been through a lot worse than you decided to deprioritize this program.
yeah, it's nothing
Yeah.
new. You're not going to hurt our feelings. We understand that it's pay to play. Like we like the patients understand you need to make money and recoup your investment as well.
Yeah.
So.
How do you think about international collaborations? Because with, I think Usher syndrome as a total category is probably between 1 in 30,000, maybe 1 in 5,000 at the very most aggressive end of the estimates. But type 1B will be a fraction of that. And so you're probably thinking US is a huge population, and obviously you start there. But how do you think about building international collaborations to cover the world?
Well, we have a Safe Site Now Europe arm. This is another family. In the rare disease space, it is hard to find motivated parents and patients who want to take an active role in finding a treatment. And when you do, you hold on to them because they're like gold and you try and motivate them and support them. And we found another family. We found other families. There aren't many, but we found like one or two. One of them happens to live in Switzerland. They're Safe Site Now Europe. And we said, listen, here's the blueprint. Here's what we're doing. Let's expand our network over there. Great. Meet new USH1B folks, build these relationships with the new scientists and researchers and biotech in that space. And then you just amplify that message and that voice. So we're also working with another organization in Australia because Usher syndrome is pretty prevalent in Australia as well. There's another USH1B group there. So the more kind of tentacles and reach you can have internationally, I think it's just better. And we are actively making sure that we are not siloed in the US here.
Yeah, why is it more prevalent in Australia?
I don't know. Great question. Yeah.
Yeah, I mean, and this is one of the other kinds of things that can be really helpful in clinical development, right? If you discover these pockets, the genetic epidemiologist in me is wondering why, but the drug developer is saying, good, that's where we'll put a site, right? This is important.
I mean, I think there's was a great researcher at the FFD found like a certain population in like in a small city in South America somewhere where like the ratio, like the amount of people to SMD was like 10 times anywhere else in the world. But it was just this little pocket. And
Yeah.
you just find these little pockets internationally. It's wild.
Yeah, interesting. Maybe you can talk a little bit about how the audience can help you. I think you have a fundraiser that's coming up. If they're in California and they want to show up to the gala, how do they do that? But also more broadly, not everybody listening is in California.
Yeah, I'll give the 30-second elevator pitch on our event. It's coming up on November 6th. It's a really fun evening. It's an evening gala. We have a live auction, a silent auction. Everyone has a great time. Sit down dinner. That's in Mill Valley, California. You can go to safesitenow.org and you can find more out about it there. And then yeah, as far as how the audience, we're always looking to partner with new research groups and new teams. Anyone interested in whether it's animal model development or it's large gene delivery systems or it's VCs who's like, I think this is a smart space to be in. Please get in touch. Those are the people we're actively pursuing right now as we build out the nonprofit and look to build new partnerships.
Amazing. Well, you and Rosalind inspire me, and I know you inspire a ton of other people the audience knows this. I lost my daughter when she was about 10 days old from a rare disease. To pick up the pieces from news like this and do something about it is amazing and inspiring. So thank you for dedicating your life to this.
Yeah, thank you, Patrick. Thank you so much.
Thanks, everybody, as always, for being with us, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.